So, okay, I will be starting my lecture today. So, I hope everyone can see my screen and my presentation is visible to all of you and I'm audible as well. So, if you have any problem, just tell me. So, good evening, all of you. Today, I'm going to present that week 5 tutorial, which is on medicinal chemistry, a course which is running on NPTEL platform. And the course instructor is Professor Harinath Chakrapani from IISER Pune. And I am Simran, your PMRF teaching assistant. I'm a PhD student at IIT Bombay. So this is week 5. I hope all of you have already gone through the week 4 tutorial and all of them week 1 to 4. So today we are going to discuss about week 5 contents. So this is the basic thing which I explain in every tutorial. That is, this problem solving session is on every Saturday from 5 to 7 p.m. and the platform is Google Meet. In case of any network disruption, please wait for a few minutes and do not leave the lecture. Please ask your queries in the chat box or raise your hand during the lecture, or you can speak it out. Please shut your mics and please switch on them whenever required, whenever you are answering something, whenever you are asking something. That would be better for us for the better understanding. So slides will be shared with the students, and your the recording of the session will be available on the YouTube channel, which is Simran NPTEL-TA. This is my YouTube channel. You can go through it, and you will find all the recordings of the session from week 1 to week 5. I have uploaded till week 4, and after today's session, I will be uploading it on the same YouTube channel, so you can go through it. And whenever you find yourself lost or you don't know what is going on, please ask questions. Please ask whatever is your doubt. I will be happy to answer it. And please make sure to attend every tutorial to understand the course better, because this is a course which requires actually time, because it's a bit hard, it's a bit confusing course, that is medicinal chemistry. So you have to be very attentive, you have to be present here so that everything would be clear to you. And before coming to this tutorial session, it is better to have that to look on that particular videos which is uploaded on the NPTEL platform by Professor Harinath Chakrapani, so in which he has already explained everything, every content, and through tutorial lectures also he had explained whatever the questions can be arise from that topic. So please have a look on those videos and then please come to this tutorial so that you will be better prepared for the questions to be asked and for the queries you will be having, so that we will have a nice chat within us. Okay. So this is all for that particular section. This is all about the things which you should know. And these are the week 5 contents, that is nucleic acids as drug targets and miscellaneous drug targets, mechanism in biological chemistry. So these are the topics which we'll be covering today in this tutorial. So questions are based on these particular content. I hope you already have seen the tutorial videos of the professor which is uploaded on NPTEL platform. So I hope I'm audible, my screen is visible. If you have any questions, anything, please ask me. So coming to after having this part, so for coming to the first question of the session. So the first question is actually the questions which was not taken on the tutorial 4. Actually, I had plenty of questions in the last lecture, so I have cut short some of the questions because of the time. So today I will be taking three to four questions which is related to your week 4, and then we will move forward to week 5 content. Okay, so starting from the question first. So here they are asking, a drug with high affinity but low efficacy is most likely a full agonist, partial agonist, antagonist, superagonist. So think about it. We have already discussed about the agonist, antagonist, and partial agonist, full agonist. We have already talked about it in the previous lecture. So it's a very straightforward question in terms of understanding. So they are asking a drug with the high affinity but low efficacy. So it is high in activity but efficacy is low. So it's most likely which of the four option you think is the correct one. So the correct answer is partial agonist. So how it is the correct answer? So partial agonist is the drug which is having a high affinity but low efficacy. It binds strongly, that means it has high affinity, but produces limited response, that means it has low intrinsic activity. So we have already discussed about this full agonist, partial agonist. So partial agonist doesn't have the potential that of full agonist. It doesn't have this high activity, but it has a strong response. So it binds very strongly, it has high affinity, but the maximum response which is produced is not up to the mark. So it produces limited response, that is low intrinsic activity. So that is why it is partial agonist, not a full agonist. Okay, this is the major difference between a full agonist and a partial agonist. I hope it is clear to all of you. If you have any doubt, please ask me. So this is the thing which we have already discussed, that antagonist and agonist, partial agonist, inverse agonist. So these things we have already talked about: the antagonist, inverse agonist, partial agonist, and agonist. We will be refreshing it today. That is, antagonist is the it binds with receptor but does not produce any effect. So it only binds to the receptor, doesn't produce any effect, any activity. And agonist, it binds with receptor and activates it very fully. So this is a full agonist. That means it will bind to the receptor and it will be activating it fully. Partial agonist, it binds with the receptor and activates it partially. So this is having a high affinity but low intrinsic activity. Inverse agonist is the agonist in which it will bind with the receptor and inhibits its intrinsic activity. So it is the inverse agonist. So as the term suggests, it is only binding it and it is inhibiting its activity. So this much we have already covered in the last lecture. So coming to the next question. So a full agonist is defined as a drug that binds receptor without activating it, produces maximum response when all receptors are occupied, reduces basal receptor activity, or D blocks endogenous ligand action. So on the basis of what we have already discussed, what do you think is the correct answer if you are talking about a full agonist? Someone wants to join? Just wait a second. So this is the question in which they are asking, a full agonist is defined as a drug that binds receptor without activating it, produces maximum response when all receptors are occupied, or reduces basal receptor activity, or blocks endogenous ligand action. So what do you think is the correct answer? Any guesses? This is related to what we have discussed in the previous lecture as well as we have discussed it today. Someone wants to answer? So the correct answer is B, that it produces maximum response when all receptors are occupied. So as I have already explained that a full agonist is have both high affinity and high intrinsic activity. So it produces the maximum possible biological response when it occupies the receptor molecule. So as soon as it activates the receptor, it shows high affinity. Good evening. It's a problem solving session. So it's a tutorial on basically whatever Professor Harina Chakrakhani has taken in the week 5. So it's a problem solving session where we will be discussing all the related questions of week 5 contents. So I hope you already have gone through the lecture videos of Professor Harina Chakrakhani, which is on NPTEL platform. So before coming to the tutorial problem solving session, you have to gone through basically on that videos, whichever like 3 4 videos are there. So you have to cover them first and then you have to come here for the problem solving session, so to understand the course better as well as to participate in the tutorial, right? So this is the focus of this particular problem solving session. So yes, please. In an easy way. See, you have to first visualize whatever the question is asking, and that will actually made it better to understand or to answer, I think. So, for to answer, see if you know the concept basically in your body, like cell membranes, polar, apolar groups. If you know that, like you don't have to memorize it. You have to understand the topic, I think. If you are understanding the topic well, I don't think that. See that though, I don't think I can answer how to memorize. But if you just repeatedly, I think, learn about it, you have a habit of actually like whenever you see the structures and how many times you are seeing it depends on that. I think you can remember it. It's not a key. There is no key like to remember it anyhow. I think how many times you are going through it, it will actually retain in your mind. I think so. So there is no actually shortcuts of remembering it. I don't think so. Anything else? Okay. So shall we proceed with the tutorial class? Okay. So okay. So here I have already answered this question that full agonist is the drug that basically produces the maximum response when all the receptors are occupied. It is having a high affinity and high intrinsic activity to produce the maximum possible biological response. Okay. So coming to the next question, when a partial agonist is given with a full agonist, it may act as superagonist, competitive antagonist, irreversible inhibitor, or enzyme inducer. So, what do you think is the correct answer? If a partial agonist, as I have already explained about the agonist, that partial agonist is having a high affinity but low intrinsic activity, and full agonist is the high affinity and high intrinsic activity. So, what do you think if you uh, combine these two, and it will, what effect do you think it will generate? Either it is a superagonist, competitive antagonist, irreversible inhibitor, or enzyme inducer. So, what do you think is the correct answer? Anyone? Anyone wants to answer? Okay. So, superagonist. No, it's a competitive antagonist. Why? Because a partial agonist competes for the same receptor but produces the less effect and reducing overall response. So basically, what happens here that your receptor is single, right? There is a single receptor in which there is a site on which partial agonist as well as full agonist is acting. So either it will attach to that or either full agonist will attach to that, right? So site is limited, okay? So if you are giving both simultaneously to a receptor, it will act as a competitive antagonist because there is a competition which will be happening between two agonists to get attached to that particular receptor. So that is why there is a competition which will be arising. So that is why there is an antagonist. Antagonist means now it will bind to the receptor but it will not produce any effect. So that is why there is a competitive antagonist which is going to be formed. So partial agonist competes for the same receptor but produces the less effect and reduces overall response. So that is why option B is the correct one. So I hope it is clear to all of you. So now coming to the next question. Yes, PDF of the okay. I will upload it on the folder. The folder is shared with you guys, I think. I don't know exactly. You will ask to the coordinator. But we have already provided the folder in which we have uploaded all these. I don't know. You have to mail the coordinator. Okay. So you people are enrolling into a course, right? If you I mean you people are enrolling into a course, right, of medicinal chemistry. So somehow you are communicating with someone to na. I don't think it's like you are just enrolling, you are just attending. Is it like that? That too you have to ask NPTEL coordinator. You have to search who is the NPTEL coordinator for your course, and then you have to mail them. You will find the email ID, I think, and then you have to mail them. I don't have the email ID. I don't know about it. Yeah, whatever it is, because NPTEL platform, we are the only ones we are taking that lectures, right? This is the problem solving session. It is apart from whatever the NPTEL courses, whatever the teaching you have been taught in that particular video lectures. This is apart from that, right? We are not connected to, because it is a problem solving session, right? You are the first. I think you are not attending the problem solving session till now, right? You haven't attended anyone. Why? I think there are two TAs which who are taking this medicinal chemistry course, right? Teaching assistant. We are only the teaching assistant taking problem solving session. We are not related to any NPTEL platform because we are just taking the problem solving sessions, right? So it is instructed to us to take this. Why is it not visible? Let me check. Let me check. Is it visible now? It's visible, right? Okay. Anything else? Yeah, but why we are discussing it here? Because this is not related to that particular thing, right? I am a PhD student at IIT Bombay. So if you are done, actually, this is the lecture in which we are going to solve the problems. So I think you haven't access to any of this problem solving tutorial either from my side or either the second TA which is there. So please be present in every problem solving session so that you will be catching up with the course, I think. Right? So it's not a good idea to skip every time. And it's week 5. I have taken already the 4 lectures. It's week 5, fifth lecture of mine. So if you understand the course better, if you want to understand it completely, please join as much lectures as you can, or I will say join every lecture if possible. Okay. So now coming to the competitive antagonist. What are these competitive antagonists? So these competitive antagonist competes with the agonist for the receptor and surmountable with increasing agonist concentration. It displaces the agonist dose response curve to the right and reduces the apparent affinity of the agonist. Basically, if you are having this antagonist replaced on your receptor, it will actually reduces the affinity of that particular agonist. So if you are giving the partial agonist and full agonist to a person, then it will behave as a competitive antagonist because now the affinity to bind to the receptor is now reduces drastically. Okay, and your curve will be. बोलिए. Yes. Actually, I have already told these things to in the starting of the session. So please be on time. It's or 5 o'clock, right? So I have already provided this thing. So you can take a screenshot of this particular thing and please be on time from next time onwards. And I'm actually discussing this PPT in every session. These points. So you can get a recording on the YouTube channel, which is Simran NPTEL-TA. You can go through that, and this recording will be available as soon as I have ended this lecture. Okay. Just take a screenshot so that there's no ambiguity left with you. So now coming to the next question. A super agonist is a drug that produces greater response than endogenous agonist, has zero affinity, is reversible, or is a always a toxic drug. So what do you mean by a super agonist? Yeah, it produces a greater response than endogenous agonist because, as the name suggests, superagonist has higher efficacy than natural ligand. So that is why it produces a greater response than the endogenous agonist. So there is a comparison table in which you can see that the property, affinity, intrinsic activity, Emax, and clinical use of all the agonists like full agonist, partial agonist, antagonist, and inverse agonist. So the full agonist has a high affinity. Affinity is present in all the four. Intrinsic activity is high for full agonist. It is moderate for partial agonist. It is none for antagonist because it actually inhibit the intrinsic activity of the receptor after binding. Inverse agonist is negative because now it will actually what it will do, it will decrease the affinity drastically of the receptor after binding it. So Emax is the curve which will be between the affinity and the intrinsic activity. So it is 100% for the full agonist. It is less than 100% for the partial agonist. And as the name suggests, antagonist doesn't participate. It is zero for that. And inverse agonist, it is below basal. So in clinical use, it is used as the activation therapy. It is used as safer activation. Antagonist is blocks the receptor. It only blocks the receptor. It doesn't produces any activity, and it reduces the basal signaling inverse agonist. So this is a table in which you can compare all these four according to the affinity, intrinsic activity, Emax, and the clinical use. So I hope it is clear. So now coming to the next question. A reversible antagonist that competes with agonist at the same binding site is called non-competitive antagonist, competitive antagonist, physiological antagonist, or chemical antagonist. So the B, competitive antagonist. So yeah, the correct answer is B, that is competitive antagonist. So what do you mean by competitive antagonist? It's bind to the orthosteric site, that is a same site as agonist. It will bind to that, and their effect can be overcome by increasing agonist concentration. So basically, this is what we have already discussed in the previous lecture. So here, the competitive antagonist is a antagonist that competes with agonist at the same binding site. So this is the agonist and antagonist. So agonists are the drugs that occupy receptors and activate them. Antagonists are the drugs that occupy the receptor but do not activate them. And antagonists only block the receptor activation by agonist. So this is the agonist molecule, and it is showing now the full activation after the receptor is binding to that agonist. Agonist and antagonist, when they are given simultaneously, there is a competitive inhibition which is happening here between the agonist and antagonist. So now there is a less activation which is possible because both of them will compete for the active side of the receptor. When antagonist is given, then it will block the receptor active site and it will not produce any activation. So this is the classical example between agonist and antagonist. So I hope it is clear. So now moving forward to the next question. An irreversible antagonist differs from a competitive antagonist because it binds weakly, it forms covalent bond with the receptor, it has no affinity, or it increases the Emax 50 only. So, what do you think is the correct answer? In which irreversible antagonist is differing from a competitive antagonist because either it binds it with the receptor weakly, it forms a covalent bond with the receptor, has no affinity towards the receptor, or increasing the curve to EC50, 50% activation. So, what do you think will be the correct answer? So, the correct answer will be the option number B, that it forms a covalent bond with the receptor. See, as the name suggests, it is the irreversible antagonist. Irreversible means it will not detach from your receptor. So the antagonist will attach to the receptor and it will detach from it. But irreversible one, it forms covalent bond. And as you know, covalent bond are harder to break. It doesn't break easily. So that is why they are irreversible. It is not, it once it binds to the receptor active site, it doesn't detach from them. So that is why irreversible antagonists permanently inactivate your receptor by covalent binding. This reduces the receptor number and decreases the activity, affinity basically. So that is why irreversible antagonist is differing from the competitive antagonist. Right. I hope it is clear. Now coming to the next question. Which type of antagonist reduces both potency and efficacy? Competitive, non-competitive, chemical, or physiological? So which type of antagonism do you think will reduces both of the potency and the efficacy towards the receptor? So what do you think will be the correct answer? So antagonism, which is reducing both. So the correct answer is non-competitive antagonism, because non-competitive antagonism reduces the maximum effect and may alter apparent potency. It actually reduces the effect of the receptor, and already if you inactivate the receptor, the potency of that particular receptor will go down. So that is why non-competitive antagonism reduces both the potency and the efficacy of the receptor. So I hope it is clear to all of you. So this is the competitive versus non-competitive antagonist. So what do you mean by these things? So an agonist, when bind to the receptor, it will produce a response, right? But what do you mean by competitive antagonist? Binding sites overlap. Basically, if there is another molecule which is present to bind at the receptor site, so there will be a competition which will be happening between the agonist, so between the antagonist and the agonist. So here the binding sites will overlap and there is no response which will be happening. And when a agonist will bind to the receptor, non-competitive functional antagonist, it differ from the binding site. What do you mean by this? That here the binding site will be different. So see, agonist is having a binding site which is correct for the receptor, right? So this is the non-competitive one. But when there is a molecule which is actually having the allosteric site, this is the allosteric site. Other than the active site, receptor have a allosteric site in which after binding to this allosteric site, the active site of the receptor changes shape. So previously it was very good for the agonist to bind at the receptor, right? The shape was exactly matching with your agonist. But as soon as your uh, this allosteric site is getting attached by this competitive allosteric antagonist, your shape of this active site at the receptor changes, which lead to the inhibition of this agonist to bind at the receptor. So now this agonist couldn't bind to the receptor active site because of the shape changes. So this is known as a non-competitive allosteric antagonist. So basically, there is no competition which is happening. Either the allosteric site is first occupying by the antagonist, and then this agonist doesn't bind to that particular active site due to the difference in now the shape of the molecule and the receptor. So this is not happening with the competitive one. So competitive one, both of them will compete for the active site, and there is a competition which will be having between the antagonist and agonist, and they are having the binding sites overlap. But in the non-competitive antagonist, what happens? The different binding site is the reason why agonist couldn't bind to the receptor active site. So allosteric site occupants will allow your shape of this active site to get changed, due to which the agonist couldn't bind to that particular receptor. So this is producing a non-competitive antagonist, right? So there is a competition between the agonist and antagonist when we call about the competitive antagonist, and there is a shape change which is happening when we are talking about non-competitive antagonist. So I hope it is clear to all of you how they are different from each other. Just look at it and try to understand. So moving forward, so now which type of antagonism does not involve receptor interaction. Is it the competitive, non-competitive, chemical, or irreversible antagonism? Basically, it doesn't involve receptor interaction. So, receptor interaction doesn't happen in which of the antagonism? Competitive, non-competitive, chemical, or irreversible antagonism? What do you think will be the correct answer for this question? Okay, so the correct answer is the chemical one. Why the chemical antagonism? It actually neutralizes the agonist directly without receptor involvement. So the chemical antagonism is the antagonism in which it neutralizes the agonist. It basically uh, uh, form reaction, basically react with the agonist only, and before the receptor involvement. So they doesn't require a receptor involvement. It chemically react with the agonist. That is why they do not involve in the receptor interaction. A chemical antagonist. Okay. So chemical antagonist neutralizes the agonist directly without receptor involvement. That is why they are not involved in the receptor interaction. They are only reacting with the agonist compound. Okay. So the chemical antagonism does not involve receptor interaction. So this is also a table for the quick revision in which we have talked about the competitive, non-competitive, irreversible site, physiological, chemical, pharmacokinetic, partial agonist, antagonism. So the mechanism Emax and EC50. So basically, you need to focus on this mechanism. So competitive antagonist has the same site and is reversible, right? So basically, two of the antagonist and agonist are competitive for the same site of the receptor, and it is reversible in nature. Non-competitive, it binds to the allosteric site, as I have already discussed that they bind to the allosteric site and they change the shape of the active site of the receptor, and due to which now the agonist couldn't bind to the receptor active site, and which leads to a thing that is known as non-competitive antagonism. It is also irreversible because once the shape of the active site changes because of the allosteric site occupants, it is not going to again change for the agonist to attach to the receptor. So now, once the change in the active site has been made, it is irreversible. It wouldn't get changed, right? So if we are talking about the irreversible one, so irreversible, as the name suggests, and we have discussed that it will form a covalent bonding with the receptor molecule, and that is why now the covalent bond doesn't break, and it will not be reversible one. It is a irreversible antagonism because in every case we are only seeing the ionic interaction and the non-covalent interaction being participating between the receptor and the agonist antagonist molecule. Whenever you are forming covalent bonds, it is irreversible in nature because they are not going to be broken so easily. So now, coming to the physiological antagonist, it is the opposite effect via different receptors. So basically, the receptor are different and it is attacking onto the different receptor. When you talk about the chemical one, so it is the chemical interaction between the agonist and the antagonist. So there is no receptor involvement which is there in the chemical antagonism. Only the agonist and antagonist will directly form the chemical bond. So pharmacokinetics is the other ADME. So this is the absorption disruption and this excretion mechanism which we have talked about in the week one. So if you have any doubt, please go through the week one video. You will find that ADME, what it really stands for, how it was happening. There is a mechanism of the pharmacokinetic system and pharmacodynamics. So these two terms we have already talked about in the week 1 lecture. So if you have any doubt, go through that video. I have explained it in very detail. So in partial agonist antagonism, I have already explained that it competes with full agonist. So the overall effect will go down. So this is a table which comes very handy when you classify these antagonists on the basis of mechanism, how they react, how the receptor binding with that particular agonist antagonist is reversible or irreversible. So this is a quick revision for that particular topic. Okay, so this is it for this antagonist and agonist part. So these are the questions which are actually left in the week 4 lecture. So I have taken up these questions now because it is related to your week 4 things. So from now we are starting the week 5 thing, and which I have already shared that these are the contents. So these are the contents for the week 5. Those of you who have joined now, please check these contents. Now, nucleic acid as a drug targets and mechanism in biological chemistry. So now we will be talking about these things. The problems will be based on now these points. So now coming to the first problem, which of the following terms best describe the programmed cell death mechanism that eliminates damaged or unwanted cell? So in your okay, necrosis. So the options are necrosis, autophagy, apoptosis, and pyroptosis. So the right answer is apoptosis, not necrosis, because apoptosis is a tightly regulated process damaged or unneeded cell without causing inflammation. It involves caspase activation and DNA fragmentation. So the question actually is asking the programmed cell death mechanism that actually eliminate damaged or unwanted cells. So programmed cell death. So here necrosis is a uncontrolled cell death that results from injury or infection, often causing inflammation. So here the programmed cell death uh, signifies of apoptosis because it is a regulated process that only removes the damaged or unneeded. Is my screen visible to you, Nikita? Can you please answer me? Is it visible now? Okay, so apoptosis is a tightly regulated process that removes only the damaged or unneeded cell without causing inflammation. And necrosis is actually a uncontrolled cell death that result from injury or infection, often causing the inflammation. So it is not a programmed cell death mechanism; it is an uncontrolled cell death. So, but here in apoptosis, you can only remove the damaged or unneeded cells without causing any inflammation, any other thing. Autophagy is also incorrect. Why? Because it is a survival mechanism where cells degrade and recycle their own components, not a programmed form of cell death. So here, cell only degrade and recycle their own components. So it is a survival mechanism, not a programmed cell death. Pyroptosis is a form of programmed cell death that is inflammatory, often triggered by infections. So they are asking only the programmed cell death that eliminates the damaged or unwanted cell. It is not going to cause anything other than the eliminating the damaged or unwanted cell. So that is why the apoptosis is a correct answer. It is only removing the damaged or unneeded cell without causing any of the inflammation. So that is why apoptosis is the correct answer rather than the necrosis, autophagy, and pyroptosis. So I hope it is clear why it is the correct answer. So now moving to the next question. The following structure belongs to a quinoline antibiotic. What is its primary mode of action? So this is the quinolone antibiotic. What will be its primary mode of action? Inhibits the bacterial ribosomes, blocks DNA gyrase and topoisomerase 4, inhibits folic acid synthesis, or it disrupts the bacterial cell wall synthesis. So, based on these particular antibiotic, what do you think? Based on the structure of this antibiotic, what do you think will be the correct one? Option A. Okay, so see what is the correct option. It is the B one. So what happens here that this quinolones and fluoroquinolones it actually inhibit the bacterial DNA gyrase and topoisomerase 4. It prevents the DNA replication and transcription that leads to bacterial death. So this DNA replication and transcription, if you want to know what they are, you will have to focus on the week 2 or week 3 tutorial, which is already uploaded onto my YouTube channel. I have already shared the name. So you have to go there and read about the week 2 or week 3 video tutorial so that you will know what is DNA replication and transcription. Okay. So as far as the question is concerned, so this quinolone antibiotic actually acts onto this DNA gyrase and topoisomerase 4 and it leads to the bacterial death. So why the option A is not correct? Inhibits bacterial ribosome. Why? Because this quinolone do not target ribosomes. It doesn't target the ribosome. It is an antibiotic like tetracyclic and aminoglycosidase do. So these antibiotics like tetracyclines and aminoglycosides, these will target to your uh, ribosome, but quinolones doesn't target your ribosome. Inhibits the folic acid synthesis. Sulfonamides and trimethoprim disrupts the folic acid synthesis, not quinolone. So, quinolone doesn't act onto your folic acid disruption. It disrupts the bacterial cell wall synthesis. So basically, beta lactams like penicillins and these molecules will target to your bacterial cell wall, and quinolones and fluoroquinolones doesn't target to your disruption in the bacterial cell wall. So why the DNA gyrase and topoisomerase 4? So why they are targeting these? So these enzymes help in DNA supercoiling and replication. As I have described here that DNA replication and transcription. So basically these enzymes, DNA gyrase and topoisomerase 4, it helps in DNA supercoiling and replication. And quinolones like ciprofloxacin bind to them and cause DNA damage and bacterial death. So these quinolones and fluoroquinolones, please remember, it basically blocks your DNA gyrase and topoisomerase. It blocks your DNA replication and transcription, which leads to the bacterial death. So uh, this is how it works. Okay. So I hope it is clear to all of you why the other options are incorrect. Now coming to the next question. The primary target of taxanes, a class of anti-cancer drug is. So this is a anti-cancer drug that is taxane. What do you think it will target on? DNA polymerase, microtubules, ribosomes, or mitochondria? So basically, it is a class of anti-cancer drugs. So what do you think in which primary target it will act on? Any guesses? So basically, if I only ask the question which are already discussed, you are not going to learn anything new here. Okay. So that is why the motivation of adding extra questions here, that you will learn something out of it. If you are only repeating the video tutorials here, some questions are actually matching and some questions are not, because the new concept you have to learn here. So if you are attending it, you should get something out of it, the new things, right? So if you answer these questions, if you know how the anti-cancer drugs will be acting, that is a new thing which you are getting here, right? It's not present on the video tutorial. So that is also a motivation of having this problem solving session. Okay. So the primary target for texanes is microtubules. Why? Texanes such as bacillotexel stabilize the microtubules and prevent their depolymerization, thereby inhibiting the mitotic spindle formation and arresting cell division in cancer cells. So basically, in anti-cancer drug, what do you target? That cell division shouldn't happen, right? It should prevent cell division. That is how you will prevent the cancer to go beyond your limit. So that is why it will target on the microtubules, not on the DNA polymerase. Why? Because DNA polymerase is inhibited by drugs like cytarabine and gemcitabine, but not taxanes. So taxanes only stabilizes the microtubules. It prevents their breakdown, and which stops the cell division. So how the cell division of the anti-cancer drug happen? It will target onto the microtubules in your body. That is how they will prevent the cell division. Ribosomes are targeted by the antibiotic like tetracyclics and aminoglycosides, not taxanes. And mitochondria, some chemotherapy drugs affect mitochondria, but taxanes specifically act on microtubules. So now you can answer here that anti-cancer drug, sorry, chemotherapy is having in the cancers, right? When you have a cancer patient, you are doing the chemotherapy to stop the cell division, right? So this is how it. If you want to define that term, please Google it, because this is a medicinal chemistry course in which I am not giving you every the definition of every single term. So this is the chemotherapy drug which is affecting on the mitochondria. If you want to know about the chemotherapy, please Google it and learn about it, how it is going to happen in the body. That will give you a better description, I think. See, I am not also not from the this biochemistry background. My background is organic chemistry, so that is why I am telling you that just if you want to have a exact definition, it's better to just Google it, because I cannot provide you a exact definition. It's a overview which I can give you how it is active. Okay. So this is how the anti-cancer drugs is targeting the microtubules rather than the DNA polymerase, ribosomes, and mitochondria. So taxanes like paclitaxel bind to tubulin and prevents it depolymerization, which arrests the cell division, which is very important to control in cancer. Okay, so I hope it is clear to all of you. Now coming to the next question, which of the following enzymes uses pyridoxal phosphate as a coenzyme? Is it the DNA polymerase, transaminases, ATP synthase, or helicase? So this pyridoxal phosphate is a coenzyme, and it is used with an enzyme known as either DNA polymerase, transaminases, ATP synthase, or helicase. So this is the answer for that. That is transaminases. So this pyridoxal phosphate is a coenzyme which is involved in amino acid metabolism. Okay. So it particularly in transamination reaction catalyzed by aminotransferases. So this pyridoxal phosphate is a coenzyme which is involved in a amino acid metabolism and which is responsible for the transaminases. So this transaminase catalyst is using pyridoxal phosphate as coenzyme to have the amino acid metabolism. So why the DNA polymerase is the incorrect? So DNA polymerase does not use PLP, it synthesizes DNA. It doesn't require any coenzyme. It synthesizes the DNA as the name suggests. Transaminases PLP is crucial for transamination, which transfer the amino group between amino acid. So transaminases do the transfer of amino groups between different amino acids, and the pyridoxal phosphate is very important for that. It is acting as a coenzyme there. ATP synthase produces the ATP but does not uses PLP. It doesn't require any coenzyme. Helicase unwinds the DNA but does not require PLP. Basically, these three things doesn't require any coenzyme to activate them or to participate in the catalyzing the reaction. But here, transaminases require the PLP, and it is very important for the transamination. So PLP acts as a coenzyme in amino acid metabolism, especially in transamination, helping to form new amino acids. Right? So this is how you will learn that this coenzyme is important for the transamination, and the transaminase is a enzyme uh, which is activating by this coenzyme. So now coming to the next question. So, what is the primary mechanism by which aminoglycosidase antibiotics exert their antibacterial effect? So, aminoglycoside antibiotics exert their antibacterial effect in which mechanism? It inhibits the DNA synthesis, it inhibits the cell wall synthesis, it disrupts the bacterial protein synthesis, or it interfere with the folate metabolism. So this is an antibacterial drug as it suggests. So how do you think it will act on which of the particular thing? Either it will inhibit the DNA synthesis first, it will inhibit the cell wall synthesis, it disrupt the bacterial protein synthesis, or interfere with the folate metabolism. So the correct option will be that C, because as the name suggests, it is an antibacterial drug. So it has to first disrupt the bacterial protein synthesis. So if you disrupt the bacterial protein synthesis, so now the bacteria will not grow in your body, because now the multiplication and the protein synthesis is now blocked. So aminoglycosides bind to 30S ribosomal subunit, causing misreading of mRNA, leading to defective protein synthesis and ultimately killing the bacteria. So if you are disrupting the protein synthesis of the bacteria, you are killing the bacteria. So that is how they are acting as a antibacterial drugs. Why it is not inhibiting the DNA synthesis? Because drugs like fluoroquinolone inhibit the DNA synthesis, not aminoglycosides. So you have to be very clear that quinolones and fluoroquinolones will act on your DNA synthesis, but aminoglycosides will act onto your bacterial protein synthesis. So beta lactams and glycopeptides, they target the cell wall. So these are the class of compounds which will act on different things, and you have to basically keep in mind while answering the question that how these things will act on different sites. So it interfere with the folate metabolism. Sulfonamides block the folate metabolism, not aminoglycosides. So these are the drugs which is acting on different things like DNA synthesis, cell wall, or folate metabolism. But aminoglycosides are the drugs that cause the error in the protein synthesis of the bacteria and ultimately kill them. Right? So aminoglycosides cause misreading of mRNA and leading to non-functional proteins and bacterial death. So that is how aminoglycoside antibiotic acts on bacterial growth. Right? So I hope it is clear to all of you how they are acting onto as an antibacterial effect. So coming to the next question, what is the key role of beta-lactam ring in penicillin antibiotic? As you all know that penicillin is an antibiotic, it has a beta-lactam ring. So now you have to answer what is the key role of that particular beta-lactam ring in penicillin. Either it binds to the bacterial DNA, it inhibits the transpeptidase enzyme involved in peptidoglycan cross-linking. It interferes with ribosomal function. It enhances our antibiotic solubility in water. So the correct answer is B, that it inhibits the transpeptidase enzymes involved in the peptidoglycan cross-linking. So why the beta-lactam ring of penicillin binds and inhibits the transpeptidase, also called as penicillin binding protein? It prevents the bacterial cell wall synthesis and it leads to cell lysis, cell death. So it doesn't bind to bacterial DNA. Why? Because penicillin do not interact with DNA. It doesn't interact with the DNA. It inhibits the transpeptidase enzyme. It is the right answer because it prevents the peptidoglycan cross-linking and weakens the bacterial cell wall. It interferes with the ribosomal function. It is incorrect. Why? Because protein synthesis inhibitors like macrolides target the ribosome, not the penicillin antibiotic. It enhances the antibiotic solubility. So the beta lactam ring doesn't affect the solubility. Okay, so that is why it is not related to the antibiotic solubility. So why inhibit the transpeptidase? Because this enzyme cross-links peptidoglycan in bacterial cell wall and inhibition leads to the cell death. So that is how the penicillin is acting as an antibiotic drug. Okay. So I hope it is clear to all of you. Now coming to the next question. The mechanism by which fluoroquinolones achieve bacterial cell death involves disrupting ribosomal function, inhibiting bacterial RNA polymerase, blocking topoisomerase and DNA guidase activity, inhibiting folate synthesis. So this is the similar question which we have already answered in the previous slides. So by which mechanism the fluoroquinolone achieve the bacterial cell death involves? So what do you think will be the correct answer? Disrupting the ribosomal function, inhibit the bacterial RNA polymerase, blocking the topoisomerase and DNA guidase activity, or inhibits its folate synthesis. So the right answer is blocking the topoisomerase and DNA guidase activity, as we have already discussed about the quinolones. So fluoroquinolones and quinolones, both of them will block the topoisomerase and DNA guidase activity, so that DNA replication and DNA transcription doesn't happen, which leads to the bacterial cell death. So, the tetracyclines and aminoglycosides target the ribosomes, not fluoroquinolones. So, fluoroquinolones and quinolones doesn't target on the ribosomes. It is the tetracyclines and aminoglycosides which will target the ribosome, inhibiting the bacterial RNA polymerase. So, rifamycins, not fluoroquinolones, inhibit the RNA polymerase. So, these drug will inhibit the RNA polymerase, not the fluoroquinolones. Inhibiting the folate synthesis, as I have already mentioned that sulfonamides inhibits the folate synthesis, not the fluoroquinolones. So fluoroquinolones and quinolones, both of them will block the topoisomerase and DNA guidase activity. So these enzymes help bacteria manage DNA supercoiling, and fluoroquinolones block them, leading to DNA strand breaks. So basically, DNA replication and transcription doesn't happen in the bacteria. So that is why the bacterial cell growth, uh, a cell that happens, the cell will not grow. So I hope it is clear to all of you. Now moving forward to the next question. Which of the following drugs is an intercalating agent used in cancer chemotherapy? It is the paclitaxel, doxorubicin, or methotrexate, cisplatin. So, which of them will be the intercalating agent? So, for that, we first need to know that this intercalation is, so this doxorubicin intercalates into DNA. So, what happens that it chelates with that, intercalates with the DNA. It disrupts the DNA replication. It will block the DNA replication and transcription, and it also generates the free radical that causes the DNA damage, making it effective in cancer treatment. So this doxorubicin is the one which helps in intercalating the DNA, and it is actually going in between the DNA, and it disrupts the DNA replication and transcription. So, what is the intercalation? So, basically, it inserts between the DNA bases. So, DNA has a base pairs. It has base pairing, hydrogen bonding between those base pair. It has a helix structure. So, this is also covered in the week 4 or week 3 tutorial, I think. So, if you want to know about the DNA RNA, then you have to go to the video lecture of week 3. I have explained in detail about that, and we have also taken some important questions over it. So you can go through that. And here, intercalation between the DNA bases happens, which blocks the replication and transcription, and which triggers the apoptosis in cancer cells. Okay. So this molecule is not incorrect. Why? It stabilizes the microtubules, but it doesn't intercalate into the DNA. Intercalation means that it inserts between the DNA base pairs. That is how you will block the replication and transcription, right? You will uncoil the DNA. So the methotrexate, it inhibits the dihydrofolate reductase, affecting the nucleotide synthesis. Cisplatin forms the DNA crosslink but does not intercalate. Right? It does not intercalate between the DNA basis pairs. So this is the doxorubicin which intercalates between the DNA basis pair, and it is used in the cancer chemotherapy. So I hope it is clear. So moving forward, so which class of antibiotics act by inhibiting bacterial folic acid synthesis? So now it will be the very straightforward question, because as I have already discussed that which are the classes which are acting on cell wall, folic acid depletion, or something like that. So we have already discussed over it. So what do you think is the class of antibiotic that is acting, that is basically inhibiting the bacterial folic acid synthesis? Is it the beta lactams, tetracyclines, sulfonamide, or macrolides? What do you think will be the right answer for the question? Okay, so the right answer is the sulfonamides. As we have discussed that this sulfonamides inhibits the dihydro pterode synthase, and it's a key enzyme in bacterial folic acid synthesis, which is essential for DNA and RNA production, and it prevents the bacterial growth. So beta lactams, these target onto the bacterial cell wall. Tetracyclines, these are targeting the ribosomes. Sulfonamides only are the targeting for the folic acid production. Macrolides, these molecule targets the ribosome again, not the folic acid synthesis. So sulfonamides are the kind of drugs which are acting onto the bacterial folic acid synthesis. And here the bacteria needs folic acid to make nucleotide basically. Sulfonamides block this pathway and prevent DNA RNA synthesis. So that is how they are acting as antibiotic drug. So basically the bacteria is not having the folic acid to make the nucleotides, which is blocked by sulfonamides. So in a way they are acting as antibacterial. Okay. So this is how you will learn that how the class of drugs will be acting on different position, different position of the uh, in human body, how they are acting, like on the folic acid disruption, cell wall disruption, either they are acting on DNA directly. So these are the class of compounds which are acting on different things. So now coming to the next question, if it is understood. So, what is the primary function of messenger RNA in protein synthesis? So, you have learned about that mRNA in protein synthesis. So, what do you think is the primary function of messenger RNA in protein synthesis? Either it serves as a catalyst for the reaction, it provides the genetic code for protein synthesis, it forms ribosomal subunit, it brings amino acids to ribosome. So, what do you think messenger RNA is doing in protein synthesis? Any guesses? So what it will do, it provides the genetic code of protein synthesis. So messenger RNA is responsible as a crucial intermediary in a central dogma of molecular biology because it transcribes genetic information from DNA and carries it to ribosome where proteins are synthesized. So messenger RNA is responsible for your genetic code in the protein synthesis. So it is not acting as a catalyst because enzyme like ribozyme or ribosomal protein catalyze translation, not mRNA. This is the messenger protein which is only carrying the genetic information from DNA to ribosome where the next protein is going to be synthesized. It forms ribosomal subunit? No, because ribosomal RNA helps form ribosomal unit, not mRNA. So rRNA is responsible for the ribosomal subunits formation rather than the mRNA. It brings the amino acid to ribosome. It is not the case because transfer RNA, tRNA, is responsible to bring the amino acid to the ribosome, not mRNA. It brings amino acid to the ribosome. So basically, here you will see that it provides only the genetic code for the protein synthesis. Okay, mRNA is responsible for the genetic code for the protein synthesis, and here you have learned that rRNA helps the ribosomal subunit formation, and tRNA is responsible for amino acids coming to the ribosome. So this is how you have learned that mRNA, rRNA, and tRNA how they are useful in protein synthesis. Okay. So I hope it is clear to all of you. And I'm just ending this lecture of today here. I hope it is beneficial to all of you. So I hope everything is understandable here. So today will be the week 4 and week 5, and we will be meeting in the next lecture of week 6. I'm ending this lecture here, and please be on time from the next time onwards. And thank you for joining. I hope you have learned something from this tutorial session. Thank you so much.